Thank you for the invite. And it's been an interesting journey this afternoon. I've really enjoyed it so far. It's, interesting. So first of all, trees are really quite resilient. And recently, around the world, and including in Western Australia, there's been an increasing incidence in the severity of tree declines. And because they're quite resilient and very long-lived, it's often very difficult to determine what the causing factors are and what are the contributing factors to these declines. So here is what we call a tree decline spiral. And we consider this as predisposing factors, which tend to be things like climate shifts, which we've been hearing about today, disturbance regime shifts. We've heard a lot about that today, forestry, mining, etc. Impoverished soils, and that's something that's very evident in the southwest of Western Australia. And often these don't lead to obvious problems, but then we have predisposing factors, and these tend to be short-term stress factors, heat, frost, and we heard a little bit from Joe, fire could be in there as well, drought, etc. And these are directly responsible for the initial decline symptoms that we see. And if it wasn't for the predisposing factors, trees would actually recover quite quickly. But they go into decline, and then we see the contributing factors. And these are the pathogens, the pests, canker fungi, and things like this. And in recent years, this has been exacerbated by the movement of humans around the world, bringing in new pests and pathogens into places like Western Australia, and so introducing exotic organisms that can cause big problems. And Phytophthora cinnamomi, Phytophthora dieback, is a good example of that, but we have many others that are a little bit more subtle. So we've been trying to untangle some of these tree declines, and we've been seeing tree declines in the chert woodlands and in rudis and a range of other species. And if we look at Gomphocephala, the chert, we've started to untangle what's going on here, and we know Phytophthora multivora, which is an introduced Phytophthora like cinnamomi, is playing a real role in helping drive that decline. Loss of mycorrhizal fungi, and mycorrhizal fungi are really important in trees, especially eucalyptus. They have a huge diversity of these below-ground beneficial fungi. Without them, they will go into decline, and I'll spend a little bit more time on them in a minute. Things like changed fire regimes we've talked about, compaction, increased nutrition, changing groundwater. These are all factors that are driving chert decline. So some complexity here, as you can see. Agonis is another one I get rung up quite recently about. People are quite concerned about the ringtail possum, for example. There are a number of things going on here too. And one of them is multivora, Phytophthora multivora is contributing to that. But interestingly, Neophysicoccum australis, a canker fungus, is native pathogen. And it's starting to starting to be a bigger problem than it was 20 years ago. Why is this? What's driving it to become a more aggressive pathogen? And it's something I would like to untangle. Rudis is another one, and we're starting to do quite a lot of work on that. And there's a range of inciting, contributing factors involved there. We've also isolated in the last couple of years a range of different Phytophthora species associated with rudis. Not too surprising, because they're on long river verges in areas that get inundated, and Phytophthora are water moles. And all of those Phytophthora species there are introduced Phytophoras. They've been introduced into Australia, and they're certainly playing a role in the decline of rudis. People say lerps, but it's not. They're contributing, but other things are also playing a role. Probably salinity as well, and this is something I'd like to untangle more in time. So I'm going to use Mary as really the story about some of the processes we use to try and understand what's driving these declines. And we have the Mary canker disease. It's very widespread. It's caused by Quambularia coiracup, and it's a native pathogen. It's evolved with Mary. Why is it now causing these massive cankers in trees? I probably get phoned once every 2 weeks about Mary decline, cankers. it takes out, produces these cankers. It basically sort of ring barks the tree under the bark, and the bark decorticates. And over time, the trees will die, as we see here. And it's quite closely related to the rust pathogen, the myrtle rust pathogen, and I'll come back to that as well. So a native pathogen, why is it causing disease? But before I go into that, why is marri important? We know it's a foundation tree species. It has a lot of benefits, and we heard a little bit from Carmen some of the things that are important about. So I'll move on. So what we wanted to do was tease out the biotic and abiotic environmental factors that might be driving marri decline, might be making this, what was an endemic pathogen that didn't really cause disease, become quite aggressive and become almost like a primary pathogen. So we established a lot of transects across the marri range to look at incidence and severity of the disease, and we were also asked, because it was also industry funded, to see if there were things we could do to control it. So here we did 62 sites across the whole range, and you can see that the red dots are severe cankers. More than 50% of the trees were cankered, and very few sites are the white dots where we saw no cankers, and they were in the drier areas. What's important with these dots is they're all along roads. It was a roadside assessment. And it was certainly also linked to loss in rainfall and temperatures as well. So climate change was playing a role there. So we also wanted to determine, okay, we're seeing a major problem along the roads, but is it actually in the forest, further into healthy stands? So we did disturbed edge, forest edge, and 50, 100, and 200 meters into the forest. And here you can see that trees with cankers, there were much more on the disturbed edge, which is a good thing, luckily, and much fewer as we went into intact forest. This is important, actually, finding. And also numbers of trees that died, much more along the disturbed edges. So, maybe an association with anthropogenic disturbances. What could those be? Pesticides, herbicides, all of those things going on. Fertilizer use, increased fertilizers moving into those forests. We know our trees have evolved on low phosphorus, for example, soils. Presence of phytophthora species and altered mycorrhizal communities. I've talked about their importance. So the questions were what are the predisposing factors that are driving the marri and the canker disease? Is it differences in mycorrhizal species diversity between disturbed sites and intact forest sites playing a role, the loss of those important beneficial fungi, and the presence of phytophthora species? So we collected soil from 17 sites across the entire marri range, and at the same time we looked at canopy height, diameter, leaf litter deposition, soil nutrition, and the presence of pesticides and herbicides, which I won't talk about so much now because we didn't have the money to do the herbicides and pesticides properly. The soil, we then grew marri seedlings in and looked at growth, the presence of mycorrhizal fungi and phytophthora in those pots. And immediately if we start looking at soil nutrition, you can see here very, very high levels of phosphorus in the disturbed sites, dropping off very quickly as we moved into the intact sites. And similar sorts of trends with these other nutrients. And you can see that here in this trial with the control, which is a potting mix optimum nutrition, the disturbed edge, we're starting to see trees growing very well, but a lot of phytotoxicity, probably from phosphorus. And then 200 meters in, a healthy plant, but a little bit smaller. Then we looked at the mycorrhizal communities in these sites. And here you can see there are three sort of discrete circles, and they're sort of separated from each other. The healthy forest is quite removed from the disturbed edge, and we've got a lot of crossover in the middle with regards to mycorrhizal communities. And if we look at mycorrhizal richness, you can see along the disturbed edge and the forest edge much less richness in species present. than in the forest. And we know mycorrhizas are really important. We lose them, then the plants are unable to take up a range of micronutrients, trace elements, etc. So they start to wind down, a bit like us. If we're not feed properly, we'll be more predisposed to disease, etc. And this is just showing sort of a relationship between some of those biotic and abiotic predictor variables that could be involved in the decline. So along the y-axis, you can see proportion of trees with cankers. And the first one, we've got a negative relationship with mycorrhizal richness. So you can see as mycorrhizal richness increases, we get less trees with less cankers. As we get more overstory diversity, we also get less cankers. And the same with nitrogen. As with phosphorus, as that increases, we get more cankers. So there's some relationships there. And we can see that again with some of the other nutrients. So copper, for example, is very important in plant defense, much less, and the very low levels of copper, which is present on the disturbed edges, for example, we get more cankers. So stories starting to evolve there. And I think probably we're going to have the same story with rudis, for example, with loss of mycorrhizas and so on. So I think there will be relationships between different species. We also isolated a range of different Phytophthora species, as you can see here. The famous one, Phytophthora cinnamomi, is quite high, but two others, Multivora and Versiformis, important pathogens, and certainly playing a role. And when we did glasshouse trials with those, you can see here the control and then the root biomass of plants which are infected with Multivora and cinnamomi. All those fine roots have been taken out in the presence of Phytophthora. And we lose those fine roots, there's nowhere for the mycorrhizal fungi to attach onto to help benefit the tree. So certainly we think these fungi are playing a role. So anthropogenic disturbance is really playing a key role in helping drive the incidence and severity of cankers in the landscape. So I suppose we need to be aware of that with regards to how we also manage our forests going further in. The more disturbance, the more likely we are to get cankers in time. And maybe as the climate gets drier and warmer, we will start to see more cankers as well moving into the forest. But that's just a hypothesis at this stage. The other thing I wanted to show was this pathogen, which is related to the canker pathogen. It's a white rust. It's also related to the myrtle rust we heard about earlier, or the eucalypt rust. It's a basidiomycete. This is an introduced pathogen. And all of that, and it takes out all the developing fruit. It likes nice succulent tissue. And this is going to impact in time on food resources like for cockatoos, I would imagine. And it's widespread. We're finding that deep in the forest. It's a primary pathogen, not just along the edges like the other canker pathogen. So this is sort of maybe an elephant in the room. So this is just revisiting that decline spiral to show that with MARI, we've started to show that fragmentation, loss of mycorrhizal fungi, the excess soil nutrients, and probably some and limited soil nutrients, Phytophthora, all playing a role in that decline syndrome. So what are we starting to do about that? I've been lucky enough to have got 3 sites planted across high rainfall, low rainfall, intermediate rainfall, with 170 families across the MARI range from 18 provenances. And we're looking at resistance to the cankers, and also working with the University of Western Sydney to look at things like hydraulic failure, drought resistance, heat resistance, and some of these other factors we've been hearing about. They're trials that are available to anybody if people would like to use them. It's a very important resource. But already we're finding evidence of heritable resistance to the blight disease. And we're now going to take those provenances to the East Coast and see if those provenances or those families that are resistant to blight are actually also resistant to myrtle rust. Sorry, the myrtle rust, the eucalypt rust. Because if we start to select for these, it'd be nice to know we're also selecting for resistant to the rust, because the rust will get here sometime, I'm sure. And MARI are known to be quite susceptible. What's interesting with the blight disease resistance, The more resistant families tend to be from the south coast, not from the northern provenances or the provenances, intermediate provinces.
Charles, what's blight disease? What do you mean by that?
Oh, that's the blight pathogen is that one. This one. The introduced one. The trials are those provenance trials in the field are surrounded by diseased cankered trees, and we're waiting for natural infection. The trees are too young yet, they're 4 years old, so the next few years we'll start to see cankers coming in, and then we'll be able to look at canker resistance. We just have to be patient. I've got to get off. Fungicide treatments, there are some things we can do with fungicide treatments, with phosphite and fungicide tilt that's beneficial for basidiomycetes. So in summary, I really just wanted to say the causes are complex. They can be difficult to determine, and these range of factors are involved, nutrient shifts, loss of beneficial mycorrhiza, the introduced pathogens. The other one is that I'm quite concerned of with warming and drying. I think we're going to see more of these opportunistic pathogens, native pathogens becoming more aggressive. And something we need to keep an eye out for. So thank you.